Hello and welcome to Nikolay's genetics lessons, and today's question is: which of the following peptides is more likely to have an alpha-helical structure and why? And here is the polypeptide chain number 1 and polypeptide chain number 2. In my previous video I said that the first thing that we are looking for is presence of the proline or glycine in polypeptide chain. If we have such amino acids, it is very unlikely that such polypeptide chain would fall into alpha-helical structure. But none of these polypeptides has these two amino acids. Before I will show you how to solve this problem, let me start from afar. Imagine that here is a wire, so one wire, and here is another wire, which represent a spring. Now imagine that you were told to use any of these wires and make structure like this. Now the question, which wire, this one or this one, you're going to use in order to make a structure like this. Most of you, I hope, is going to choose this wire because it's going to be energetically more easier to make a structure like this than if you use, for example, this wire in the form of spring, because probably first you have to straighten it, maybe even apply a heat and maybe use a hammer, and then you can bend it in a such structure. And it is much easier to use this wire. So we thinking about using one wire, another wire, in order to make a structure like this from the point of view of energy that we have to spend, maybe time and effort, in order to make a structure like this. And believe me, evolution is also using the same principles. Evolutionary, our cells are not going to use higher energy cost products if we have other low energy products in order to make a desirable, in our case, protein. So now we are going to consider both these amino acid sequences from the point of view how much energy is needed to make alpha-helical structure. The less energy we need, the more probable that this sequence or this sequence is going to be used in order to make such helical structure. Scientists already have found statistical data which we need in order to do such calculations. And we say that, for example, alanine has the highest helix propensity, and excluding proline, which we usually never find in the middle of the alpha helix, glycine has the lowest, approximately 1 kilocalorie a mole less favorable than alanine. So alanine would be 0 in our table and glycine would be 1. And do you remember that glycine is very flexible, most flexible amino acid, and it's unlikely that we can find it in the middle of the polypeptide chain. But glycine still can be found here, but proline is probably cannot be found. It is even absent from this table because proline, very rigid structure, disrupt hydrogen bonding in alpha helices. So we can consider every amino acid from the point of view of energy required for its integration in alpha helical structure. Now let's compare both sequences. For example, sequence A of amino acid, and each letter here stands for one amino acid, start with LKA, and this sequence also starts with LKA. So we even don't have to look for each amino acid in this table because both this polypeptide chain would have the same energy requirements. But this polypeptide A, next amino acid number 4 is E, and amino acid number 4 in this polypeptide chain is A. E stands for the glutamic acid and A stands for the alanine. And as you see, alanine has 0 propensity and glutamic acid, according to our table, has 0.4. Let's put on top of this E 0.4, 0.4, and on top of this A 0. So far, these two amino acid chain are different in this amino acid. Let's check NDE and AE. So here we have D, and D stands for the aspartic acid. Let's check our table here. ASP is going to be three-letter abbreviation of this amino acid, and it is 0.69. So let's put it also on top of this letter, 0.69. So let's round it to 0.7. And in amino acid chain B, we have again alanine here, which is 0. Now let's continue comparing these two polypeptide chains, EAA, EAA, RAM, RAM, and here we have S. S stands for the serine, which is in our table is 0.5. So let's also put 0.5 here. And in polypeptide chain B, we have here alanine, again, which is 0. Propensity is 0. E in polypeptide chain A is glutamic acid. We already know that this is 0.4, 0.4. And here we have M, which is methionine. And let's check methionine in our table. It has 0.24. Propensity 0.24. One more time, if we compare these both sequences are going to be the same, except these amino acids which are underlined, and those which are underlined in polypeptide chain A require more energy than this in polypeptide chain B. This means that polypeptide chain B, its amino acid in this sequence has highest alpha-helix propensity, and it is more likely that this polypeptide chain would make alpha-helix. It is the same like we compare with this wire, this sequence, and this sequence we can compare with this wire. It would be much easier to use this one to make a structure like this. And for our cells, from the point of view of energy conservation, it would be easier to use this sequence to make an alpha helix than this one. And this is all for today. Subscribe and see you in the next video. Goodbye.